Okay, nice, welcome. So it's our last class before the long weekend, last class before a bit of a break. As I mentioned earlier, these classes are new for me. This is just a new set of classes about genomics and so on. I don't know how far we'll get today in our slides. We'll just pick it up next week. So don't worry about that. We'll just see how it goes. And also, the other thing to not worry about is term test marks. So our term test marks are posted. I'm sure most of you know that. Probably many of you didn't do as well as you had hoped. I just want you to see the big picture. In this course, like what, over 35% of your grade comes from skills and from quizzes, not things that aren't terribly discriminating, in which you're doing very well. So I think if you use the grade estimator and put in those marks, you'll realize that overall in the course, you're doing pretty well. The final average for this course likely will be close to 80%. That's almost as high as is legal. At this university, like the Senate isn't all that excited about averages over 80. But we're going to be really close to that. So we're kind of doing as well as we can as a group. But as individuals, you don't care about the group. You only care about your own record. And what I'm trying to say is you have lots of time to improve your record in this course. The final exam is like next year, the 27th or something. It's like a long time from now. You have lots of time to prepare, and that's 30% of your grade, so lots of chance. Anyway, come to office hours if you have some concerns, post to the forum, usual thing. Dennis and I will help you. If you're a crumb, then now's the time to act. Okay. You live in a country where transgenic goats are legal. Yeah, awesome. Transgenic goats are legal for making human therapeutic proteins. And transgenic humans are legal, sort of. It seems really obvious that if we can make a transgenic goat that makes spider silk protein in its milk, surely to God we can help some people who suffer from genetic diseases. Surely there's some kind of gene therapy that we can invent to help people. And that's been so obvious for 30 years, but we've not made a lot of dramatic progress. It's a bit like cancer. A lot of people working on it, some breakthroughs, but just not a lot of general progress. And of course, one of the problems is that the number of disorders that we could hope to help is relatively small. If you're going to try to cure someone's genetic disease, well, it would need to be a single gene disease. It's very difficult for us to try to cure some complex disease that's controlled by 50 genes. But if you have a disease because of a lack of function of a particular gene, then maybe we can help you by genetic engineering. Maybe we can make you a GMO. There just aren't all that many of those diseases. And we're not good enough at inserting the gene in exactly the right place yet. And we're not very good at regulating the expression of genes. I mean, that's the thing in eukaryotes, right? Their genes are highly regulated, And we're just not very good at that. So we're kind of restricted to diseases that are single gene and for which we can just stick a bunch of protein in. So here's a list. I'm never going to ask you for this list, but for your information, these are some of the kinds of diseases. So severe immune deficiencies, you might have heard of bubble children, kids who have to live inside a sterile bubble because they have no immune system. That's a single gene disorder in many cases, and maybe we can help those bubble boys by inserting this gene. We tried that, but with some tragic results. So again, this whole area is improving, but it's troublesome. So in general, for human gene therapy, then there are these two kinds of strategies. One is here on the left, insert some gene. Let's say we're trying to help people with cystic fibrosis. Single gene disorder affects your airway, your digestive tract. Let's say we're going to try to deliver CFTR to the airway cells of these people with a viral vector that infects airway. I mean, this example is liver, but doesn't matter. So we've tried that, and we've had some transient success at delivering CFTR into the airway tissues of affected people. It just doesn't last very long. So using a viral vector to infect the host. Another way, of course, is to take cells out of the sick person, and this works better for blood disorders. Take bone marrow stem cells out of a person, genetically engineer those cells, and then put them back into the person. Take the cells out, engineer them or put them back in, and then put them back in. Again, some success and some tragedy in trying to do this with blood disorders. And then the third route is to use embryonic stem cells, engineer them, and then insert them. So all of these strategies have been tried, limited success, but lots of promise. And I'm sure that this will be part of your lives as adults. Gene therapy will be a thing for certain disorders. A more sort of radical kind of gene therapy is about to happen with the creation of 3-parent offspring. This, to our knowledge, this has not happened yet, but it has been approved, and families have been selected. And I suspect, certainly before you graduate, and maybe before next summer, the first human will be born with 3 parents. This is gene therapy in a grand scale for people, essentially for women who carry mitochondrial disorders, who carry mutations in their mitochondria. When women create eggs, when they create gametes, they put mitochondria into those gametes. Males do not, mostly. You might get a little tiny bit of mitochondria from your father, but it's almost all from your mother. So mothers who carry mitochondrial disorders, all of their children are affected. So various kinds of strategies here could be used to take a nucleus from mom, sperm from dad, fertilize them, and then put that into a different egg. Put that into a different oocyte that has normally functioning mitochondria. So the baby that arises has a mitochondrial mom and a nuclear mom and a nuclear dad. Has 3 parents. This seems to work okay in animals, but we're going to give it a go for the first time in humans soon. 3-pair babies, kind of a radical kind of gene therapy. This is very, very significant because the children born of this procedure now will be cured of that disease. And all of their offspring will be cured, and all of their offspring will be cured. We've never done that before. We've never messed with the genome, with the genetics of people that will be passed on forever. That's a big ethical barrier. In this case, I'm sure that those kids in the future would be grateful that they no longer suffer from this disease. But what if it wasn't a disease? What if we just didn't like people with red hair? And we made sure we didn't have any kids with red hair. Maybe in the future they wouldn't appreciate that. We shouldn't be making decisions for people in the future, people who don't even exist yet. So this has been a kind of a thin edge issue where we are now making decisions for people in the future, but we're pretty sure they would be okay with that. If you want one of these, it's only $12,000. It's a clone because the person who originally had this cat, the cat passed away, they were very sad. They had $12,000 extra. So they cloned their cat because they couldn't bear to lose the original. So this is a clone. And so Viagen, this is real. I mean, you might think you're sitting in some kind of sci-fi movie right now looking at Viagen. It sounds very futuristic. This is a real company. It really does clone your cats for $12,000. It'll clone your dog for $50,000. It'll clone your dog. It'll clone your horse. You have a champion horse, you can clone that sucker. If you have a champion dairy cow, you can clone that. Cloning animals is a thing. It's legal. It's a little expensive, but I would hope as first-year biology students you would have some sense how this is a bit futile. So you clone your dead cat. You don't get the cat back. You get a different cat. It's a clone, but it's not the same cat. Come on. In what way is it not the same? Anyone? How it grows up, so its environment as it grows up. All the epigenetic changes that happen, an even bigger one.
Memories and experiences.
It doesn't have the same memories and experiences. What's a good genetic thing that's different? It'll. Is it going to die a lot faster because it was cloned from adult cells? Maybe, probably not. We'll come back to that. What's one way that if I cloned this woman, her clone would be not the same at all? You're trying too hard. Grew up somewhere else? Yeah. Genetics, biology students, this woman's going to be not the same at all as her clone because methylation patterns. Get out of the second term. Go to the first term. Recombination, her gametes would be different. Yes, her gametes would be different after recombination. How's she going to be different? Okay, argue over lunch. Look, Barbra Streisand, $50,000 to clone a dog, she cloned 2. So, I understand Barbra Streisand is not even maybe recognizable to your generation. Substitute the celebrity of your choice. When celebrities start doing this, everybody wants to do it. Everybody wants to do it. So this is going to be a thing, cloning your pets. Don't do it. It's futile. You will not get the same creature. So we've been able to clone like 22 different kinds of animals, but we couldn't clone a monkey. We couldn't clone a primate until now. And this is one of the things I love about this course and love about biology, because fundamental things are changing all the time. Now we can clone monkeys. And this is a technique called somatic cell nuclear transfer. This is the same technique that was used to make Dolly, like that originally cloned sheep. That was years ago. Somatic cell nuclear transfer. So we've been doing that. That's how we cloned the cats and the dogs and the horses and so on. But it would never work with a monkey. This is the process. So take, in this case, fetal skin cells. So that's relevant. These are fetal skin cells, these fibroblasts. And we take a donor egg from this top monkey on the right, take a donor, and we pull the nucleus out of that donor egg. Pull the nucleus out of the donor egg. And then we can sort of smush those two cells together. Fuse them together. The fibroblast and the oocyte, fuse them together. And by a little bit of magic, we can activate that, and it thinks that it's been fertilized. It thinks it's been fertilized. And this egg begins to develop into an embryo using the nucleus from this fetal fibroblast. We tried this for years and it kept failing. And now these researchers have discovered, okay, it's an epigenetic thing. We've got to mess with the epigenetics. So we put in this stuff, KDM, blah, blah, blah. This is a histone demethylase. So that detail is not important. What's important is that we do some epigenetic modification of this embryo. We block HDACs, We put in some demethylase. So we help this nucleus get over the epigenetic barriers so that, where's Fatty, that question? So they don't die earlier if we can modify them epigenetically. Then we transfer that embryo into a mother, and we get a cloned monkey out of that. And in fact, we got two. This diagram is a little confusing because it colors all the monkeys pink, and it's hard to keep track of them. But this little clone monkey, these clone monkeys, their genome came from this skin cell. They are clones of that donor skin cell. They're not clones of their oocyte mother. They've got mitochondria from their oocyte donor, so they've got a mitochondrial mom, but the surrogate, they have no genetics, no genetic inheritance from the surrogate mother. All of their genomic DNA has come from that skin cell. All of their genomic DNA has come from that skin cell. This epigenetic treatment has been the barrier that has been preventing success in cloning monkeys. The efficiency of this process is still horrible. Horrible. But now we at least know what we have to do. We have to figure out what are the epigenetic barriers and help cells get over those in order to clone. So we know it's possible. And if you can clone that animal, you can clone that animal. If you can clone a monkey, you can clone a human. I think we all know that someone's going to do that. somewhere there is a society, or at least a little micro-nano society of people who will try to do this. In your lifetime, this is going to be, cloning humans is going to be a thing. Maybe it won't be that person wanting to make a copy of herself because she likes herself, like she's pretty awesome, so why not make another one? It might not be that kind of cloning, but it might be this kind, where I take some kind of stem cell from you, I pull the nucleus out of that stem cell and put it into one of those eggs. If you're a woman, maybe one of your own eggs. And as we just saw with the monkeys, we can get that to grow into an embryo. Then we can take those embryonic cells and grow them into whatever we want. So if my kidneys are failing, maybe I can clone myself, not to make another whole person, but enough to make new kidneys for myself. Make new kidneys. So we'll call this therapeutic cloning, where I'm making a clone of myself, I'm making this embryo, but I'm not going to grow it into a person, I'm just going to grow it into some organ that I need. Or maybe I would grow it into a person, because who could resist that? That embryo right there, at least in theory, could be implanted into a cooperative woman who wouldn't mind carrying this clone. Lots of people will do that. So, again, you will need to deal with this. Maybe you'll want to clone yourself therapeutically. In Canada right now, human cloning is illegal. You can't clone humans in Canada. All of this stuff we've been talking about, messing with genomes and creating special cells and genetic machines and clones and all of that, of course, what's at the root of that is making life itself. The human mind will not resist that temptation to create life from scratch. People are trying right now. They will continue to try, and in your lifetime they will succeed in creating living cells from non-living pieces. And depending on your definition, that's already happened. This SIN1, these cells, it's a bacterium called mycoplasma. This bacterium, SIN1, is called Cynthia in the literature, Cynthia. This is the first organism in the universe, the first organism in the universe that did not inherit its genome from a previous cell. These are the first cells in the history of the universe. In the history of the universe, the first cells that did not inherit their genome from a previous cell. These cells got their genome from a computer. These researchers sat down at a computer and typed out the genome, put it together, put it into an empty cell, and booted up that empty cell. An artificial genome put into bacterial cells. And it worked. So this was, by one definition, the first synthetic life ever. A cell that did not have a genome. We pulled the genome out of a cell, and then we put in our completely artificially constructed genome, and it worked.
So they used pre-existing cells to do
Used pre-existing cells, swapped out their genome for our custom-made genome, and we were so arrogant we even put a little bit of poetry in that genome that we put inside that somebody who was doing sequencing in the future could discover. There's all kinds of other information coded in that genome. In order to create life from scratch, you need to know what the basic pieces are. You need to know what is the minimal genome. What is the minimum number of genes? What are they that can support independent life? So, if you were just beginning to be a graduate student and that was your project, where would you start? What would you do to try to figure out what's the minimum set? What's the minimum genome? An idea. What would be useful? What would be, yes?
Find the organism with like the smallest genome.
Beautiful. Find the organism that's got the smallest genome ever and see what genes it has. Great. Keep going. That's one strategy. Find the smallest genome ever. You won't be shocked to hear that mycoplasmas seem to be those creatures. Yes. Okay, keep going. How do you know which genes are essential? Find genes that are expressed really strongly in all cells. Is that what you were going to say? Find something that's common from all cells, and that's probably important, probably essential. Okay, those are both observational studies. So there's observational studies. Which organisms have the smallest genome? Which genes are common to everywhere? And then the genetic strategy, knock out genes one by one until you discover the ones that are essential. Wonderful. Those are all terrific ideas. Very creative approaches. That's essentially what people have done to try to discover how big the minimum set is. And I'd like you just to think for a moment, maybe write down, like, what do you think the minimal set is? Do you think like 50 genes would be enough to make a cell that would live on its own? Or 500? Do you think cells might need 5,000 genes in order to be? So if you were trying to make a cell from nothing, would you have to make 500 genes? Just think about what that would be. Because now I want to tip out of sort of DNA tech per se and creating cool stuff and move into more genomic analysis. I notice on the back channel that someone says, going back to the clone question, don't we suffer all kinds of environmental mutations as we grow? Absolutely. So as you grow, your cells suffer mutations. So clones would be different in the mutations that they suffer over the course of their lifetime. But there's still one huge difference that I didn't hear that is peculiar to females. It has nothing to do with the mitochondria. Good. X inactivation would be completely different in females and their clones. Lovely, lovely. All right. Back in the first term. I kept saying how envious I was of you becoming young scientists in a world where sequencing is just no longer an issue. Man, when I was a grad student, sequencing a little piece of a gene was horrific. Radioactivity everywhere. You could only do a few bases at a time. It was a nightmare. So people of my generation are really, really excited that now sequencing is just so easy that you don't even have to think about it. But I want you to sort of understand in general how this works, because it's such an important part of genomic analysis. And I've shown you these kinds of data before, how the cost of sequencing is dropping like a rock. And I want to give you some basic understanding of how does this happen? How does a technology like this change so rapidly, so dramatically from one year to the next? So let's talk a little bit about sequencing. If you are going to sequence a genome, at this moment, our abilities are still limited to fragments. So we still need to break the genome up in pieces, and then we sequence the pieces, and then we put everything back together. So we fragment the genome, we sequence the pieces, and then we reassemble those fragments back together. And if you studied DNA sequencing in high school, it probably was this. It probably was what's called Sanger sequencing, after Fred Sanger, who invented this and discovered and got a Nobel Prize for it, because it was, like, again, such a revolution to be able to sequence DNA. So this can be a little bit tricky. The first concept is that sequencing DNA is synthesizing DNA. All of the techniques that are used today to sequence DNA are actually using some kind of DNA polymerase. Sequencing, they're not actually determining the sequence of a template directly. They're asking, what is the sequence of the complementary strand that I'm synthesizing? That's what we're actually doing. So this strand to be sequenced at the top, we're not actually determining which base comes first and second and third. We are determining which base is being added to the 3' end. We're determining which bases are being added to this 3' end of this primer. Of course, we create this reaction tube that contains the nucleotide we want. The nucleotide, in this case, we'll just say ATP. But we put in a very small amount of this alternative nucleotide called dideoxy. Dideoxy, look at the difference. No 3' OH. Dideoxynucleotides have no 3' OH. And now you can predict what will happen. If there's no 3' OH, then you will not be able to add the next base. DNA synthesis will stop as soon as you add a dideoxy base. So this is a mix that's mostly normal nucleotides, but contains a very small amount of dideoxy. So every time a dideoxynucleotide gets inserted, the synthesis will stop. And it will stop. So what's very, very important here for you to understand is that all of these pictures, they always show one template, but there are billions. Billions of templates are being replicated in each of these tubes. Billions of them in each of these tubes. And some of them, watch this, You see this first C? In the tube that has the CTP, the dideoxy CTP in it, some of the templates that are being replicated will stop right there. They'll put a dideoxy C in right there. And as a result, the fragment will be quite short and it will travel quite far by electrophoresis. But in some of the other templates, a normal C will be put in there and a dideoxy C will be put in next. And that fragment will be a little bit longer and not go so far. In other templates, these C's will both be normal. And then this first C down here will be dideoxy. So that fragment will be longer still. So in the tube, billions of molecules being replicated at once, some fraction of them get stopped, get interrupted at various places, yielding fragments we can separate on a gel. This is the technique that explains why sequencing was expensive. This is labor intensive, takes a long time. In the old days it used radioactive markers. Now we're using fluorescent markers. But one of the advantages, one of the innovations, one of the inventions was to do this all in one tube. Oh, I forgot this slide. This is just reinforcing that the dideoxynucleotide will cause termination of synthesis because it has no 3' OH. But one of the ways that this became much cheaper is because it became automated. So that's one of the answers. Why is this so much cheaper? Is it became automated. We have DNA sequencing machines and robots that do all of this in one tube by using fluorescent nucleotides. And because we have fluorescent nucleotides, we can run this all out on a very small gel, and a laser can ask which base is at the end, and we can read off the DNA sequence. So huge warehouses full of DNA sequencing machines going night and day. So on the back channel, someone was saying, so why we have this drop in sequencing costs? This is one of the explanations. Automation. Someone had to invent this machine. Thousands of careers for you in industries developing scientific technology, lots of it. Jobs you've never even heard of before. Someone had to invent a PCR machine. Someone had to invent these DNA sequencing machines. It could be you. If you have any interest in chemistry, after sitting in chemistry all morning, then put those together. Put biology together with chemistry and be the person who invents the next machine and patent it and get rich. Because we always need to do this faster. And one of the technologies that has made a huge difference is called next generation sequencing. Next gen sequencing. And look, massively parallel. Massively parallel. The problem with Sanger sequencing is that we can only do one fragment at a time. Like in each tube, there's only one fragment being sequenced. One at a time. Now you could have 1,000 tubes, or you can have like 1,000 machines, but you're still just doing one fragment at a time. But look at this. We're just skimming the surface of this. I don't expect you to understand all the text. I'm going to use the word magic a couple of times. But the principles are the same. Take a genome, bust it up into pieces, and stick these linkers on the end. And you want the linkers on the end because that's what holds your fragment onto a solid plate, a solid surface. So no test tubes anymore. None of those little plastic tubes that you're working with. No. A glass plate with thousands and thousands and thousands of these fragments stuck onto one plate. That's so important. On one plate, we've got thousands of different fragments. Thousands of different fragments on one plate. The fragment that was originally stuck at that location here in the bottom corner, it gets amplified by PCR magic. So it was only one copy of that fragment, but then it got amplified by PCR to make thousands of copies of each fragment. So on this glass plate, we've got thousands of fragments and thousands of copies of each. That's the massively parallel part. Massively parallel part. But it's still based on synthesis, because we determine the sequence of these fragments by asking which base gets added to the 3' end, to this 3' primer, which base gets added. And again, we know that by fluorescence and lasers and magic. But this is another reason why the cost of sequencing has dropped so dramatically. The invention of massively parallel sequencing robots that can now sequence simultaneously thousands of fragments all at the same time. Nice. Okay, so those two things, automation and massive parallel, those are the big developments that have helped us do this really quickly. And who knows, you might invent the next one, the next big breakthrough in DNA sequencing that makes it simple. I don't know, does your generation watch Gattaca? You should. It's old, but it's new. This young woman, she meets a guy in a bar, she thinks, maybe I like him, maybe I don't. In 10 minutes, she's got his DNA sequence to figure out whether she's going to continue this relationship or not. You might invent the technology that generates DNA sequences in 10 minutes for people to use in bars. Watch Gattaca over the weekend. This slide just reinforces that idea that we're sequencing pieces of a genome, and then we ask the computer to find how all those pieces overlap. And sometimes students get confused about this, like how could these pieces possibly overlap from one genome? It's one genome, but it's, again, billions of copies of that one genome. Many, many, many copies of the genome get all broken up into pieces. We sequence all those pieces, and then we rebuild the sequence by noticing how the pieces overlap. So the third contributing factor to the drop in price is computing power, our ability to invent computers to deal with this data. So if you have any interest at all in computer science and biology, man, we need you to figure out how to make sense of these data. Because if you can sequence stuff that easily, then look what people do. You just go into an environment and scoop up everything. From the ocean floor, from your intestine, from soil, from the mouth of a fruit bat, from the slime at the bottom of whatever, you just scoop everything. You extract the DNA from an environment. You extract the DNA from an environment, not from isolated organisms. That's what we've been talking about up to this point. You extract the DNA from a whole environment, bust it up, sequence it all, put it back together. This is called metagenomics. Finding the DNA in a whole ecosystem. Because if you're making synthetic cells, you're going to want to do that. You're going to want to make a product. You're going to want to say, oh, you know all that plastic that's floating around in the ocean? I invented plastic-eating life. I got the plastic-eating gene when I scooped up all the soil from some swamp in the Amazon. I have no idea what organism that gene came from. I don't care. I was mining the biosphere. I was mining for genes. And I found this gene that degrades plastic, and I put it into my synthetic cells. And now, if you want to buy that from me, we could talk. We could talk. You can imagine where this is going. People building engineered genetic machines for specific purposes and becoming extremely wealthy as a result. Metagenomics, crazy idea. What we're discovering, of course, metagenomics is ridiculously powerful because those of you who are interested in microbiology, the whole field of microbiology, and for that matter, the whole field of virology, is based on organisms that we can culture in a lab and study. Everything we know about microbiology is pretty much derived from those few species that we can culture in a lab. But this approach allows us to pull genes from organisms that we don't even know exist. We don't even know that they exist. The plastic digesting gene might have come from some virus that infects octopus that we've never even discovered yet. But we've got the gene, and we can put it into our synthetic cell and make a product. Someone had a question. Yeah?
How do you know what protein or function that gene can produce?
How do you know what protein or function that gene can produce? You clone the gene, you put it in a vector, and then if you're looking for plastic-eating genes, you put all the genes you can find in vectors, and you say, can you degrade plastic? Can you no. Oh yeah. So you just screen, you just look for the ones that can do the activity that you want. All right. So we've got to the sequencing part, and when I see you again, we'll talk about that.